This is part two of the chapter 35 lecture notes. This is where we left off. Um, so on this slide, you see the four main types of neurons. The one that we looked at um, on the previous slide, here we go, is called a multipolar neuron. And a multipolar neuron has a cell body with many dendrites extending from the cell body at one end, and then an axon, a single axon, extending from the cell body at the other end. A unipolar neuron has a single axon, and then a bipolar neuron has, and by the way, bipolar disorder, you hear that so much. Bipolar neurons have nothing to do with that. So, a bipolar neuron just has dendrites at one end and an axon at the other end, but there's the dendrites are not directly connected to the cell body. There's a fiber that connects the dendrites to the cell body as well. And then a pseudo-unipolar neuron has dendrites and axon that are continuous, that don't actually connect directly to the cell body. So the just like anything in anatomy and physiology, this is like the theme of our class. If you ever take anatomy and physiology, the structure and function are always linked. In fact, we always say structure determines function, or the anatomy determines the physiology. So the structure of the nerve cell determines its function, um, whether it's multipolar, or even if you go back and look at a multipolar neuron, um, the entire structure determines its function. All of these branches are receiving signals, and then this long fiber is like an electrical wire. It's shaped like an electrical wire. It acts like an electrical wire. Um, and then this looks like an electrical circuit, which is exactly how it acts. It's exactly how it behaves. So, picture A is called a pyramidal cell of the cerebral cortex. The cerebral cortex is the outer layer of the cerebrum, which is the largest area of the brain. That when we say our left and right brain, we're talking about the cerebrum. Purkinje cell of the cerebellar cortex, which is the cerebellum, is if this is the uh, I don't draw very well, but if this is the cerebrum, the cerebellum is a part that's kind of here. It's not the brain stem, but it's kind of posterior or dorsal to the um, well, it's closest to the spinal cord. That's easiest to say. But anyway, it's a Purkinje cell of the cerebellum. And then olfactory neurons, which are olfactory means they function in smell. Okay, so we've talked about neurons a little bit and their diversity. Certainly haven't gone into a lot of detail, but at least you can see that there are different types, and their structure determines their function. So they have play different roles in the um, nervous system. Glial cells are sometimes called neuroglia. There are different types of glial cells or neuroglia. They are for support, protection, and nourishing the neuron. They outnumber the neurons 10 to 1 in the brain, and most brain tumors are actually caused by mutations in the neuroglia or the glial cells. So, a few different types. A microglial cell functions in uh, defense of, pathogens and that kind of thing. An astrocyte in the central nervous system, in the brain, functions in maintaining the blood-brain barrier. So, in other words, it keeps blood vessels and the blood itself from contacting the neurons in the brain because the blood carries, along with nutrients, it also carries toxins and sometimes even bacteria. So, it's very important to keep the neurons from becoming infected because once you lose it, it's gone. The neurons don't have the ability to regenerate. Ependymal cells produce the cerebrospinal fluid, which is CSF, cerebrospinal fluid. Oligodendrocytes, we already mentioned, produce the myelin that wraps around axons, acts as insulation, and causes the nerve impulse to travel faster down the axon. And then, okay, so that was the central nervous system. In the peripheral nervous system, which is, like we said, the central nervous system is the brain and the spinal cord, and the peripheral nervous system are the nerves outside the brain and the spinal cord. So, satellite cells perform the same role as astrocytes in the central nervous system. They keep the neurons from contacting the blood. And Schwann cells perform the same role as oligodendrocytes. They produce the wrapping, the myelin around the axons. So here are the notes on the glial cells. In the central nervous system, oligodendrocytes are the ones that form the myelin sheath or the insulation around the axons. The astrocytes are the ones that form the blood-brain barrier, and I know what it says here, but that's what they do. The ependymal cells produce the cerebrospinal fluid, and the microglia are for defense, so they help protect against pathogens and toxins. And then in the peripheral nervous system, Schwann cells perform the same role as oligodendrocytes. They form the myelin, and satellite cells perform the same role as astrocytes. They protect the neurons from the blood, from contacting the blood. Okay, so when neurons communicate, the signals are possible because each neuron has a charged cellular membrane or a charged plasma membrane. It's still a cell, so that the outside of a animal cell is the plasma membrane. And the charge is a voltage difference between the inside and the outside. So you call that voltage, or um, a, a it's actually called the resting membrane potential. The resting membrane potential is the voltage across the plasma membrane. It's kind of like a battery has a positive and a negative end. The outside of the plasma membrane is positive and the inside is negative. So if the charge changes, if it reverses to where the inside is now positive and the outside is now negative, then that allows for the that voltage change to spread throughout the neuron, and um, that results in a nerve impulse. So the charge, the voltage can respond to neurotransmitters, chemicals, serotonin, dopamine, um, norepinephrine, are some common ones, acetylcholine. Um, these neurotransmitters are chemical signals, and it can also change in response to a voltage change or a mechanical change. So these are images of voltage gated sodium channels. And what they do is it's just one of those channel proteins inside the plasma membrane. You can see this is a plasma membrane. Remember, it's kind of review, but you can see from Bio 111, the plasma membrane is a phospholipid bilayer, and we learned that there are different proteins scattered throughout the plasma membrane that play different roles. So this is just a channel protein that has a gate on it. You can see the gate right there. Okay, so that gate can be opened by a voltage change. So if the voltage changes, um, the outside of the cell we talked about was positive and the inside is negative. But if that voltage changes to where the inside becomes less negative and more positive, that can open the gate. So that gate opens and these sodium ions that are on the outside can move in, and sodium has a positive charge, so it makes that inside that was negative, it makes it become more positive. After a while, you don't want a nerve impulse to just continue indefinitely, so after a while, something will cause the inactivation. So there's this little um, structure called the inactivation gate that will come in and close off, um, that will shut off the channel and won't allow any more sodium in. And so the membrane potential won't become any more, the membrane voltage or potential won't become any more positive. So down at the bottom, voltage-gated ion channels regulate the concentrations of different ions inside and outside the cell. And these ions are charged particles, and when charged particles flow, that's electricity. So the flow of ions is the same thing as electricity. It is electricity, and that's what a nerve impulse is. It's just electricity or flow of ions that travels down the axon of the of the neuron. The difference in total charge between the inside and outside is called the membrane potential. So at rest, the resting membrane potential is negative 70 millivolts. And so what happens is that resting membrane potential is created and maintained. Actually, it doesn't say on here, but if you remember from Bio 111, we talked about the sodium potassium pump. Well, the sodium potassium pump, oops, sorry, the sodium potassium pump pumps 3 sodium ions out of the membrane for every 2 potassium ions in. So you can tell that, like, if this is the outside of the neuron and this is the inside, there's going to be more positive charges on the outside and more negative on the inside. So because 3 sodium ions would be more than 2 potassium ions. Um, so the sodium potassium pump maintains the resting membrane potential in neurons. And then that resting membrane potential can be changed by different stimuli, a stimulus from a sensory cell, a sensory receptor, or um, another neuron can depolarize the target neuron to its threshold potential. And depolarize means that this outside that was positive and the inside that was negative, creating that negative 70 millivolts. Well, depolarizing means that that negative 70 becomes more positive. So it might become negative 60 or negative 50. You know, it would go in the positive direction. And when it's depolarized to its threshold potential, which is somewhere around negative 55, depending on which textbook you read, um, then lots of sodium channels open. Sodium rushes into the cell, and the neuron depolarizes to about positive 40. So it goes from negative voltage to positive 40 millivolts. And at that point, um, that's as high as it would go. Just want to click on the figure and see